Bem-vindos ao nosso espaço, um mergulho profundo nos assuntos mais interessantes de esportes, ciências e temas gerais do momento. Aqui você vai encontrar conteúdo de qualidade, com análises empolgantes e descobertas fascinantes que vão expandir seus horizontes. Nossa missão é trazer informação relevante de forma dinâmica e descontraída. Queremos que você faça parte dessa jornada incrível de conhecimento e diversão. Se gostar do nosso conteúdo, não esqueça de deixar seu like e se inscrever no canal para não perder nenhuma novidade. Ative o sininho também. Aproveite!
If you love science, don't forget to like and subscribe. When we think of Alan Turing, we usually picture the brilliant mathematician who broke the Enigma code, laying the foundation for modern computing. But Turing had another, far stranger, passion: biology. In 1952, he published a paper titled The Chemical Basis of Morphogenesis, introducing one of the most elegant and profound concepts in biological science: the Turing pattern. Imagine a fertilized egg. It's essentially a homogeneous, uniform sphere of cells. The paradox is this: how does something perfectly uniform spontaneously decide to generate complex, ordered patterns? Like the spiraling shell of a nautilus, the intricate veins of a leaf, or, most famously, the stripes on a zebra? Turing proposed that these patterns weren't governed by a massive genetic blueprint dictating every single spot, but rather by the simple interaction of just two chemical substances, which he called morphogens. This mechanism is known today as the reaction-diffusion system. Think of it as a chemical race involving two distinct participants: the activator and the inhibitor. First, the activator. This morphogen is the pattern maker. When its concentration reaches a certain level in a cell, it encourages the production of more of itself, essentially accelerating the process. It's like a positive feedback loop. More activator means more pattern. Crucially, the activator only diffuses slowly and locally. It stays close to where it's generated. Second, the inhibitor. The inhibitor's job is simple: to counteract the activator. When the activator starts building a pattern, the inhibitor is simultaneously produced. However, the inhibitor has a fundamental difference: it diffuses much faster than the activator, and it spreads globally. This difference in diffusion rates, the fast inhibitor versus the slow activator, is the entire engine of biological patterning. Imagine the skin of an embryo. A small fluctuation causes the activator to spike in one tiny region. This spot starts to darken, or stripe. But as the activator rises, it quickly triggers the production of the fast-moving inhibitor. The inhibitor races away from the central activated spot, creating a chemical buffer zone, or a protective ring, around the activated area where no other activation can occur. It suppresses the pattern-making process in the surrounding tissue. The result? A dark spot, surrounded by a sterile, white, or unpatterned ring. When the inhibitor's concentration eventually falls far enough away from the initial spot, a new spike of activator can safely emerge, repeating the process. This controlled spacing is what gives rise to patterns of regular wavelength, whether they are stripes, spots, or mazes. This elegantly simple mathematical model explains why we see such regularity in nature. The pattern you get, stripes or spots, is entirely dependent on the asterisk scale of the tissue relative to the wavelength of the reaction-diffusion system. Take the zebra and the leopard. Why stripes on one and spots on the other? It's often about the size of the area being patterned. If the tissue is very long and narrow, like the leg of a zebra, the system naturally favors stripes. Imagine wrapping the pattern around a cylinder. The spots merge into lines. If the tissue is wide and flat, like the main body of a leopard, the system favors isolated spots. The genius of Turing's work is its universality. The same underlying differential equations that describe the pigments on a zebra's skin can also model the spacing of hair follicles, the growth of certain plant root structures, the organization of brain folds, and even the oscillating chemical reactions seen in laboratory experiments, like the Belousov-Jabotinsky reaction. The implications are staggering. It suggests that while genes provide the instructions for making the chemical components, the activator and the inhibitor, the physical patterns themselves are not encoded line by line in the DNA. Instead, they are spontaneous emergent properties of physics and chemistry interacting over time. Biology, in this sense, is simply solving a math problem set by the conditions of the initial tissue. Alan Turing never lived to see the full impact of his biological work confirmed through modern genetic and cellular analysis, but his legacy persists, showing us that the complex tapestry of life is woven from surprisingly simple mathematical threads. For more incredible facts like this, don't forget to subscribe and turn on notifications.